Antigen is a foreign macromolecule which can initiate an immune response inside any host. Any large macromolecule can serve as an antigen, including those in bacteria, viruses, protozoa, helminths, food, snake venom, serum components, red blood cells, or the cells or tissues of various different types of species. The antigen structure does not match any structure of the host, that is self. Therefore, antigen in majority of cases is a non-self structure. B cells or T cells present in the host identify these non-self structures as foreign and mount immune response against it. Our immune system has designed various mechanisms for protecting our body from diseases. The immune system requires some hint from the invading pathogen to mount immune response against it. Cells of immune system recognize the invading pathogens by specific non-self structures present on them. These non-self structures are some macromolecules present on invading bacteria, viruses, fungi, or any other parasite, which are known as antigens. As soon as the presence of the non-self structure is registered by the host body, immune response is mounted against it. As this non-self structure leads to the generation of immune response, it is also known as an immunogen. An antigen is basically a marker of any invading agent for a host cell, which can be used to identify its foreignness. These antigens are a part of the basic structure of any organism. These are the integral structures of the organism in which they are present. These structures will not be considered as antigens for the host, but they will act as antigens for any other organism. Every organism possesses some structures inside the cells or on the surface, which can be identified as markers or antigens on these cells. Blood group antigens in human beings, feather antigens in birds, cat hair antigens in cats, sheep RBC antigens in sheep, and bacterial antigens are a few examples. The structure of outermost part of the bacterium may contain some motifs which can function as antigens, which are unique to the microorganisms. They are somatic antigens, like antigens which are present on the cell wall of gram-negative bacterium are known as somatic antigens. They are designated as O antigens. Capsular antigens, the antigens in the bacterial capsule are the capsular antigens. Flagellar antigens, antigens present on the flagellum of an organism are known as flagellar antigens. They are denoted as H antigens. Protein substances such as exotoxins and endotoxins produced by pathogenic microorganisms are also antigenic. The terms antigens and immunogens are sometimes used synonymously, but there is a slight difference between them. Immuno-gen, a macromolecule which can generate immune response. Immunogens can bind to the antibodies and generate the immune response, whereas antigens can only bind to the antibodies. Now, the difference between immunogenicity and antigenicity. Immunogenicity is the ability of any macromolecule by the virtue of which it can generate the immune response and also bind to the antibodies developed as a result of the response, whereas antigenicity refers to the binding of antigens to their complementary structures, the antibodies. Now, we will talk about the antigenic determinants. An antigen is usually a complex macromolecule folded in a specific pattern. Immune cells interact with the antigens at restricted portions of the molecule. These differential small motifs can bind to different types of antibodies and activate different clones of B cells or T cells. These structural subunits are the sites where the antibody actually binds an antigen and are known as epitopes or antigenic determinants which bind to the antibody's paratope or complementarity determining regions. Antigens may possess more than one type of epitope. They can have multiple epitopes of same type as well as different types. The epitopes of antigens identified by T cells or B cells are different. The epitopes identified by B cells are usually on the surface of the antigens and may be three-dimensional. T cells recognize only those peptides which are exhibited on self-MHC, the major histocompatibility complex molecules. These peptides may be from the embedded portions of the protein antigens which are exposed only after processing or cleavage. Exogenous and endogenous antigens. Exogenous antigens are the antigens which enter our body from external sources. They enter our body during various infections such as bacteria, viruses, fungi, protozoa, helminths, etc., or environmental substances such as dust, mites, foodstuffs, pollens and pollutants, etc., by inhalation, ingestion or injection. The immune response developed against these antigens protects us from their adverse effects. The endogenous antigens are internally generated molecules due to the normal cell metabolism or due to intracellular bacterial or viral infections. These antigens are presented on the cell surface associated with class 1 major histocompatibility complex. Some examples for endogenous antigens include self-antigens, tumor antigens, alloantigens and some viral antigens. Now, we will talk about the factors governing the immunogenicity of a macromolecule. Immunogenicity of any macromolecule depends on many factors. The possibility and extent of immune response generation also depends upon the macromolecular architecture. Properties governing the immunogenicity of any macromolecule are the molecular size, foreignness, chemical nature, heterogeneity and susceptibility to antigen processing and presentation. First, we will talk about the molecular size. A macromolecule can generate an immune response if its molecular size is high. Efficient immunogens usually possess molecular weight above 10,000 daltons. Molecules with high molecular size activate the B cells efficiently. Small antigen molecules are unable to cross-link the B cell surface receptors. Therefore, its stimulation is also prevented. Hemocyanin or serum albumin is more immunogenic than insulin or glucagon. Some low molecular weight macromolecules exhibit antigenicity but not immunogenicity. They are known as haptens. Haptens are the molecules which can bind to antibodies but cannot elicit the immune response. They are low molecular weight macromolecules which cannot stimulate the B cells or T cells to mount the immune response. Immune response can be generated against them by binding small size haptens to some carrier molecules. These carrier molecules are usually proteins. The antibodies developed against this conjugate may be against hapten molecule, carrier molecule and the regions binding the hapten carrier complex. Some drugs, peptide hormones and steroid hormones can function as haptens. Now, the next part, foreignness. Immune response of our body depends on the degree of variability between self and non-self structures. This variability is foreignness. The foreignness increases with the increase in evolutionary difference. Immunogenicity increases with increasing foreignness because the variation in the macromolecular structures increases. For example, an immunogen from worms can elicit potent immune response in human beings as compared to an immunogen from the goat. Goat is also a vertebrate and possesses close evolutionary relationship with humans as compared to worms. Microorganisms possess greater evolutionary divergence, therefore, they stimulate our immune cells with great efficiency. Next is the chemical nature. Chemical composition of macromolecules has an important role in contributing towards their immunogenicity. Proteins are the most potent immunogens amongst all biomolecules. After proteins, polysaccharides also serve as effective immunogens. Cells of our immune system are less responsive towards lipids and nucleic acids. Immune response to most of the infectious microorganisms is towards their protein antigens. Proteins are highly immunogenic because of their large macromolecular size and diversity in their structure. Next is the heterogeneity. Heterogeneity in the macromolecule enhances its immunogenicity. Macromolecules composed of same repeating units are weakly immunogenic, such as homopolymers, example starch or cellulose. Natural heterogeneity exists to the highest levels in proteins. Therefore, majority of the antigens are proteins. Proteins naturally contain different types of amino acids, which accounts for complexity in the structure. Complexity further increases because of their primary, secondary, tertiary and quaternary levels of structure. Heterogeneity in the molecular structure further increases by complexing two types of biomolecules. For instance, glycoproteins and lipoproteins may be more immunogenic than proteins. Immunogenicity of polysaccharides and lipids can be enhanced by conjugating them with other biomolecules. Next is the susceptibility to antigen processing and presentation. Generation of immune response against certain antigens requires their digestion into smaller units. The protein antigens, which can be engulfed by antigen presenting cells, that is APCs, are cleaved into polypeptides. These short stretches of polypeptides can be presented to the T cells, which in turn will lead to the immune response. Antigens which cannot be digested and presented by antigen presenting cells are poor immunogens. Next feature of an antigen is specificity. Specificity of an antigen is its property to bind to a specific antibody only and not others. The antibody raised against an antigen binds to it by the complementary structure. The binding will occur only if the attachment sites on antigen and antibody exactly match each other. In very rare cases, the antigen binds to an antibody whose structure is close to its complementary structure. This feature is known as cross-reactivity. The specificity or cross-reactivity of any antigen is the attribute of structural similarity or dissimilarity in the macromolecules involved. The specificity or the property of an antigen binding to the specific antibody is due to the compatibility in their binding sites. If two different antigens possess some bit of similarity in their structure, they may bind to same antibody. This can be further understood by classifying antigens into various categories. Types of antigens: organ specific antigens, species specific antigens, autoantigens, alloantigens and heterophile antigens. First, we will talk about the organ specific antigens. The organ specific antigens are the antigens associated with a particular organ of all the members of the same species. These antigens are associated with the given organ and not found on other tissues. Example, thyroglobulin is specific for thyroid. Structural units of any given organ in all the members have same function. For example, all the nephrons of the kidney perform same function of the blood filtration. Therefore, these cells possess similar structure for performing the same specific function. Nephrons of all the members of the same species will have same structure, therefore same antigens. This structural similarity accounts for presence of organ specific antigens. These antigens can be the markers of that particular organ. It may also be present on the particular organ in even unrelated species, example adrenal antigens and kidney antigens. The next is the species specific antigens. These antigens are the markers of a particular species. They are present in all the members of any given species and are not present in other species. These antigens help in distinguishing the species immunologically. For example, serum albumin of horses is immunologically different from that of the man, dog, sheep and so on. These antigens can also be helpful in tracing evolutionary relationships. Next, we talk about the autoantigens. Sometimes the immune system identifies the self structures as foreign and mounts an immune response against it. These structures are the autoantigens. These antigens are the self antigens of any host, but due to some reasons these are identified as foreign by the immune system. For example, are lens protein antigens. Cell mediated or humoral immune response is mounted against these antigens, which lead to the destruction and in turn the destruction of the self tissues. Recognition of self structures as foreign is observed in many disorders named as autoimmune disorders. For example, multiple sclerosis, myasthenia gravis, pernicious anemia and rheumatoid arthritis. Next is the alloantigens. These antigens are present in some members of any given species. The cells of some members of a given species carry one form of the antigen and others carry different form of the same structure. This difference is due to the presence of different alleles in different individuals. They are genetically determined. These antigens can be used to categorize the members of the species. For example, blood group antigens. These antigens are of clinical importance. Major histocompatibility antigens, the MHC antigens, also fall under this category. They are also sometimes termed as isoantigens. Next is the heterophile antigens. These are the antigens of similar nature present on organs or tissues of unrelated species. These antigens may be structurally closely related, but may occur in different classes or even kingdoms. Example, Forceman antigen is a lipid carbohydrate complex widely distributed in humans, animals, birds, plants and bacteria, etc. It is present on red blood cells of sheep, horse, cat, mouse, chicken, kidney cells of guinea pigs and pneumococci. These heterophile antigens have useful applications in diagnosis of diseases. Example, Weil-Felix test for typhus fever, where patients suffering from rickettsial disease produce antibodies which can agglutinate some non-motile strains of Proteus. And next example is the Paul-Bunnell's test for infectious mononucleosis, where patients suffering from infectious mononucleosis due to Epstein-Barr virus produces antibodies which agglutinate sheep erythrocytes. Now, we will talk about the biological classes of antigens. Immune response to antigens is mounted by cell mediated as well as humoral immunity. Cell mediated immune response is generated by presenting peptides by self cells on MHCs to T cells, and humoral immune response is generated against antigens by the stimulation of B cells and eventually the production of antibodies. There are two mechanisms for antibody production through humoral immunity. Antigens can be classified on the basis of the mechanism they follow to generate immune response. Some antigens provoke the B cells directly to produce antibodies, and others require the help of T helper cells. We can classify the antigens into two types on this basis: T cell independent antigens and T cell dependent antigens. First, we will talk about the T cell independent antigens. These antigens do not require the involvement of T cells for the production of antibodies. These antigens have repeating units of epitopes which bind to the B cells receptors and stimulate them, for example, bacterial lipopolysaccharides. As a result of stimulation, B cells proliferate and differentiate into plasma cells, which eventually produce antibodies. Some of the B cells are stored as memory B cells for further protection against these antigens. The next category is T cell dependent antigens. Some antigens require the help of T helper cells for the production of antibodies. They are processed by antigen presenting cells, the APCs, and presented to T helper cells. The large size protein antigens are engulfed by the antigen presenting cells and cleaved into small peptides. These peptides bind to MHC and are presented on the surface of APCs. The T cell receptors, TCRs, on the surface of T helper cells bind to the foreign peptides on self MHC and get activated. These T helper cells stimulate B cells for the production of antibodies. Such antigens are known as T cell dependent antigens. Next is the super antigens. The super antigens are a special category of antigens which stimulate a large population of T cells non-specifically. They are potent T cells mitogens, which means that these molecules have enormous capacity to stimulate the multiplication of T cells. These antigens do not stimulate the T cells against some specific antigen, but only initiate its non-specific proliferation strongly. These activated T cells exhibit massive immune response and produce large amount of cytokines. This non-specific exaggerated immune response is harmful to our body and it causes shock and multiple organ failure. They can be of bacterial or viral origin, for example, exotoxins produced by pathogenic streptococci. Now, the role of biological system in immunogenicity. The host plays a major role in generation of immune response. The immune response depends on the genetic makeup of the host, the dose of incoming antigen and its route. First, we will talk about the genotype of the host. The antigen given to two different hosts generates different levels of immune response, and the antibody titer may vary greatly in both the individuals. This is influenced by the host's genotype. The cell mediated and antibody mediated immune response depends on the recognition of antigen molecule. The antigens are recognized by either B cells receptors or T cell receptors. The B cells identify the antigens and produce antibodies against them. If the antigenic stimulus exhibits high degree of foreignness, the production of antibodies by B cells will be high. The T cells are stimulated by the peptides presented by self MHC. The degree of T cell stimulation and activation depends on the sequence of peptides presented by MHC. which in turn decides the extent of immune response. Next is the dose. The immune response also depends on the dose of antigen entering the host. If the number of antigens entering the host is very high or very low, then immunological unresponsiveness or tolerance develops. The efficient response also requires repeated doses of antigens, which enhances the number of activated B or T cells. Next is the route of administration. The route of administration of the antigen has a profound effect on the generation of immune response. The antigen entering through the intravenous route may not serve to be immunogenic, whereas its intradermal entry may elicit enormous immune response. Now, we will talk about the adjuvants. Adjuvant is derived from the word adjuvare, which means to help. Adjuvants help in enhancing the immune response. They are administered along with immunogens and are non-immunogenic. Adjuvants increase the immune response by releasing the antigen slowly. They also stimulate the macrophages non-specifically and enhance inflammatory effects. The addition of adjuvant increases the effective time of antigen exposure to immune system from a few days to several weeks, which helps in enhancing the immunity to a great extent. They can be organic compounds or can be of microbial origin. Friend's adjuvant is commonly used for effective antigen administration. Friend's complete adjuvant is a preparation which contains heat-killed mycobacteria and muramyl peptide, a component of mycobacterial cell wall. Friend's incomplete adjuvant is water in oil emulsion containing antigens in aqueous solution, mineral oil and an emulsifying agent. Aluminium potassium sulfate, that is alum, is also a commonly used adjuvant. Applications of immunogenicity. The antigens are also used as vaccines. The property of antigens to generate immune response is applied for the preparation of vaccines. Vaccines are killed or attenuated preparations of microorganisms. The antigenic preparation without the presence of toxic components of pathogens is developed to immunize the host against this pathogen. There are numerous different types of antigens on the bacterial surfaces. A single organism such as a bacterium may contain a variety of proteins, carbohydrates and other materials that can provoke the immune response. Now, concluding the topic, the immunogens are foreign macromolecules which can induce the immune response of any host. All organisms possess some structures in their cells or on their surfaces which can function as antigens. Immunogenicity of any macromolecule depends on its size, chemical nature, complexity of the molecular structure and degree of foreignness. Large macromolecules like proteins, glycoproteins and lipoproteins are highly immunogenic because of the complex nature. Antibodies bind to the antigens at particular site known as epitopes. Immune response inside an organism is influenced by its dose, route of administration and genotype of the host. Immunogenicity is applied for preparations of vaccines which provide immunity to the hosts.